Hi guys, Mr. Kennedy again. This is the second part of your Chapter 1 lecture notes. We're going to learn a little bit about the scientific method. By now you've probably already seen the video on the scientific method, maybe have some graphic notes of your own. In case you missed anything in that video, I want to give you an opportunity to get a little of that information from me. So thought I'd run you through the steps of the scientific method, and by the time we're done, hopefully you'll have those down and be able to tell the difference between a hypothesis, theory, and a principle. So we're going to start like this. First step, scientific method. Scientific method always starts with an observation and a hypothesis. Now, I know what you're thinking, Mr. Kennedy, I'm not a scientist. I will never use the scientific method. Well, just in response to that, I'm going to put a little question out there and just ask, what would happen if you lost your cell phone? I would hope that you would start looking for it, right? And as you started looking for it, you might start to predict the different places that you might have left your cell phone. Guess what you just did? You just used the scientific method. I lost my cell phone as an observation, and you trying to figure out where you left it, you're making hypotheses. Those are educated guesses. So you use the scientific method a lot more than you probably realize. Observing and hypothesizing, it's just one way that scientists can answer a question, generate new questions, solve problems. That's what the scientific method is all about. So the reason we use scientific method is because from one science to the next, we want a set of common steps that we can use to problem solve. That adds validity to any claim that we might make in response to an observation or a problem that we're trying to address. If we just went at answering problems, answering questions with any kind of process, as opposed to one that's common from science to science, someone could argue that the results that we get were only because of the way we went at answering the question, right? That's what's called experimental error. So that's the one thing that we want to avoid. So having a common set of steps helps eliminate error in the experimental process. Biologists use the scientific method, but so do chemists, so do physicists, so do geologists. Any area of science you could think of, they use the scientific method. So again, it starts with an observation. Once that observation is made, we go on to a hypothesis. Hypothesis is an explanation for a question or problem that can be formally tested. First thing you need to remember about a hypothesis is that definition. It has to be something that can be tested. So I'm going to put it out there. Is the hypothesis black cats cause bad luck something that can be tested? If you're shaking your head yes, then I would really like to know how you're going to test the concept of luck. The reality of it is black cats cause bad luck. That is not a good hypothesis, because what is luck? How do we define the word lucky? What is lucky to one person might be unlucky to someone else. And I always use the lottery for this one when we're talking about luck. If I won the lottery, some of you might think I'm really, really lucky. If I won the lottery and I was constantly being pestered by people asking me for money, some of you might look at me and feel sorry for me, right? How do you define luck? So a hypothesis, first of all, has to be something that can be tested. Second, it has to be unbiased. We can't put our opinion in it. So one of the things that you never put in a hypothesis when you write it are the words I think or I believe. We don't put those words in there because as soon as you do, you've told us that you've kind of already made up your mind what the outcome should be. I think it's going to be this. I believe it's going to be that. Hypotheses cannot have the words I think or I believe in them. In junior high, you probably learned to write a hypothesis using the words if then to prevent that sort of bias. Once you have a hypothesis, the next step would be to conduct an experiment. Experimentation is the process of testing your educated guess. Now keep in mind that an experiment does not prove anything. It is a test to help us draw a conclusion of whether or not a hypothesis is correct. So no matter what type of experiment you're doing, the data that is produced does not prove your hypothesis to be correct. It will either support your hypothesis or it will lead to continued testing. Now, how we set up the experiment is something that is very special. In experimentation for this class, I'm going to introduce you to the type of experiment called a controlled experiment. Controlled experiments are experiments that have two groups associated with them. One group is called the control group, and the other group, the experimental group. The control group is the group where all of the conditions in the test that's run on this group are kept the same. So basically, this becomes our benchmark or our baseline for us to use as a point of comparison as we run tests on our second group, the experimental group. So the control group is the group in which all of the conditions are left alone or in as natural a state as possible. The experimental group is the test group. This is the one that we run our experiment on, or we apply what's called our variable to it. So when you run an experiment, let's say, for example, I want to know the effects of alcohol on muscle coordination. So I might take a group of rats and ask those rats, or make those rats, run through a maze. And as those rats are running through the maze, I'm going to time how long it takes for them to go through the maze. Well, since rats don't normally drink alcohol, the control group in my experiment would be a group of rats that I asked to run through the maze, but all I give them is water to drink. The experimental group would be the group of rats that I asked to run through the maze, and as they run through the maze, they are given alcohol to drink in whatever quantity that I see fit in terms of my experiment. So that group that gets the alcohol, that's the experimental group. The amount of alcohol I give them, or the alcohol itself, that is the variable that is being given to that experimental group. All right, now, in terms of those variables have names. There's a variable in an experiment called the independent variable. The independent variable is the only variable that actually affects the outcome of the experiment. And the way I like to describe it is it's the one that I'm in control of as the researcher. So in my example that I gave you, the alcohol that was being given to the rats is the independent variable. That's the one that I am giving and I am in control of as the researcher. There's a second variable, it's called the dependent variable. The dependent variable changes in conjunction with the independent variable. So as I, the researcher, control the independent variable, whatever I do with the independent variable is going to cause a change to the dependent variable. So in kind of a complex way of saying it, the dependent variable is called the dependent variable because it depends on the independent variable. Another way to look at it that's more direct and very straightforward, your dependent variable is basically your data, right? So in my experiment that I gave you with the rats, the alcohol was the independent variable. The time it takes them to run through the maze, that's the dependent variable. The more alcohol I give them, theoretically, the more time it's going to take for them to run through that maze. Now, in my experiment with the rats, that type of experiment is a controlled experiment, but there are some experiments that we do that don't have a control and an experimental group. Those types of experiments that are not controlled are basically referred to as a biological investigation, or basically, observational experiment. Those types of experiments are the ones that are usually done when we're doing field work. Like I might do something where I'm counting how many birds fly by in a migratory pattern. There's no experiment there. I'm simply charting or taking observational data. In the event that I go back to my controlled experiment, my experiment is going to give me some data. So the next step in the scientific method is the collection of data. So we'll just jump to that. The information obtained from our investigations is called data. That data can come in two forms. It can be numerical or it can be descriptive. When you gather numerical data, that type of data, whether it's time, temperature, length, mass, area, volume, as you see here, that type of data is what's called quantitative data, because you're getting a quantity of something. If the data is expressed in a verbal form, observational data, then it's qualitative data. So if it's got numbers in it, like time, temperature, length, all that, quantitative, that's a quantity of something. And if it's just descriptive, hey, the liquid turned purple, that's qualitative data. Okay. Now, using that data, it's very important that we tie it back to our hypothesis and come up with some sort of conclusion. That's the next step in the scientific method. We need to ask ourselves, did our data support our hypothesis? If it did, great, we're ready to go on to the next step. If our hypothesis was not supported, then the next step would be to go back to your hypothesis, revise it, and start over. Okay, so if we go to the cell phone example, if you were thinking, gosh, my cell phone might be in my backpack, but it wasn't there, would you just stop and go, well, I guess it's not my backpack, and I still don't have a cell phone? Or would you revise your hypothesis and go, well, let me look somewhere else. Let me look in my pants pocket, my pants that I was wearing yesterday or something. So you go through the dirty clothes and you look there, right? You would keep looking until you got to your cell phone. So once your data supports your hypothesis, the next step is to verify your results. This is a complex process that would involve your data being peer reviewed, but for now, I'm just going to put it to you this way. Your hypothesis, once supported by data, will have to be tested again by other people. In order to be considered valid, those people have to be members of the scientific community, and they have to come up with a similar result as what you did. If this happens, and it happens repeatedly over long periods of time, you can get your hypothesis elevated or graduated up, if you will, to a theory or a law. A theory is an explanation of a natural phenomenon that's supported by a large body of scientific evidence obtained from many different investigations and observations. Laws are basically the same thing as a theory. It's just that they've been tested even longer and in greater depth and detail. Once we get to theories and laws, your results will be reported out into scientific journals and generally accepted in the scientific community to be known as something that is true and fact, like, I don't know, the law of gravity, so to speak. All right, gang, so that's the scientific method in a nutshell. We're going to be spending a lot of time using it throughout this year. So I hope you enjoyed these notes, and I will see you next time.